All right, so now on to me. I thank the previous speakers for setting up a lot of stuff for me so that I don't have to repeat it. So I'm going to divide this talk. Actually, before I get into my objectives for today, a couple important disclosures. I am a co-inventor on a patent for some of the monoclonal antibodies I'm going to describe today, and I do consult for a company that's working, this ID Biologics, that's working on getting this antibodies into EV-D68. But before I launch into the actual talk, just to let you know what we're going to do. So I'm going to have two main parts. The first part, I'm going to talk about a recent study of a human autopsy from a patient who had AFM due to EV-D68 and highlight what that one unique case has taught us about the pathogenesis of this virus, of how it causes disease. Then I'm going to switch gears and go into monoclonal antibodies, which I talked about already at the previous iteration of this conference a few years back. But I'm going to give you some updated data and really focus on two antibodies that are kind of in various stages of development to hopefully get put into humans one day. And at the very end, try to tie all of that together into what I think of as my current model for how EV-D68 causes AFM, which builds, again, on all these other talks as well. And then where I think antibodies might present an option for therapeutics, and give finally a progress update on one slide on how we are progressing. So, all right. EV-D68, not a controversial statement to say that it's a cause of AFM, but that wasn't true for a number of years. So while we knew this is a sort of a snippet of the incidence of AFM graph that others have already shown, we knew that when EV-D68 peaks were happening in 2014 and again in 2016, as represented here, there was a lot of EV-D68 circulating generally at those times. And then a number of the patients who had AFM would have EV-D68 isolated from their respiratory tracts, typically, although from other sites as well, including very rarely from the cerebrospinal fluid. And then cases in between of AFM that happened in these outbreaks, we never really did see EV-D68 in those cases. So there was definitely an association between the virus and the disease, and a lot of work that I don't have time to talk about using different mouse models of disease, investigating the presence of enterovirus antibodies in the spinal fluid of patients with AFM, but not other patients with other types of neurologic disease. A lot of evidence built over the course of years to really suggest that EV-D68 isn't merely associated with AFM, it causes AFM. And you just heard a couple of talks that indicate some ways that EV-D68 may in fact be doing this. So I'm going to talk about, though, a case of an autopsy from an actual human that we were able to get some samples from and really try to learn a little bit about this virus. So this is a case from 2008 is when the patient was actually infected with EV-D68 and had AFM. And this was a 5-year-old boy. And this story will sound pretty familiar to all of us. But remember, this all happened well before 2014, before we were using the term AFM. This patient had a common cold, just like a bunch of his classmates did. And then about a couple days later, started to have some weakness. This weakness was asymmetric. It was in his upper extremity. Also had a little bit of voice change. These are all things that we would commonly see at the presentation of AFM. And then over the course of just another 48 hours, that progressed very rapidly and significantly to the point where all of his limbs were affected. He was incontinent, and then eventually the muscles that control his breathing failed. So this would be considered a very severe case of AFM. And unfortunately, that's when this patient passed away. And some astute clinicians realized at that time that, boy, this looks a lot like poliomyelitis, and we really shouldn't be seeing poliomyelitis anymore in the US. So we should, they pursued an autopsy, and they did actually find EV-D68 in this patient's cerebrospinal fluid. So that is a little bit different. Well, that case was published in 2011. And again, it was really in 2014 where this idea that EV-D68 and AFM were linked really firmly set in, and then all the research since then. So we reached out to these investigators and were able to pull out the samples from their autopsy that had been fixed in formalin and preserved in paraffin, and take a closer look now that we sort of understood the significance of this case. And I will say the other unique aspect of this case is that, most patients do not die during the acute phase of AFM. In fact, most patients don't die of AFM at all, because these days we're very good at supporting people who have respiratory failure and people who can't, eat because their swallowing muscles aren't working. And so this may be one of the very few cases where we do have the opportunity to do such a study on someone who unfortunately does pass away in this, what I would consider acute phase of AFM. So what did we find? So this is really the main finding for sure. What you're looking at here, this is a section of this patient's spinal cord, and you're seeing consecutive sections. So basically we took a section here, and then this is the very next section. And on the left, we stained for viral RNA. And so this is showing very beautifully sort of a neuron cell body and one of these axonal structures. And this is in the patient's cervical spinal cord, which correlates to kind of where the motor neurons are that innervate the arms, the upper limbs. And then on the right, we see nice staining. In this case, it's immunohistochemistry showing nRVs D68 capsid proteins. And since these are back-to-back slides, this structure correlates with this structure, and this structure correlates with this structure. So you're seeing in these same structures, we see lots of viral RNA and lots of viral protein. And just to prove that we're not just cherry picking the best example, we did see a lot of examples of this where we saw what looks morphologically to be neurologic structures, or neuronal structures that have this staining for both of these RNA and protein. We also did further investigate the tropism, because it really seemed to be neuron specific, and that was indeed the case. So this is, again, still spinal cord samples, 2 samples going across the top and the bottom, serial sections, where we stained for that viral RNA, and then we also stained with what is called NUN or NOYN, a neuronal marker. And we saw that nRVs D68 infected these structures that had the viral RNA were indeed neurons, but not every neuron was infected in this region. And it was very striking that it was just neurons. The tropism seemed very neuron specific at this stage in this patient's infection. And I show this one example where that wasn't the case, because it really was the exception that seemed to prove the rule. So in this case, we saw good RNA staining, what looks to probably be a macrophage based on morphology, but certainly didn't have this co-staining with the NOYN marker. And not only did we see all this evidence of the virus infection, but there was a pretty strong inflammatory infiltrate around these infected sections of spinal cord. So we saw lots of macrophages in those infected areas. We also saw a lot of T cells, not so many B cells. And then when we investigated which kind of T cells, these were all CD8 T cells. So these are those cytotoxic T cells. And their job is to come into infected tissues, recognize cells that have something in them that's foreign, so a virus, and to destroy those cells, to not let the virus take a hold. That's normal function. Well, when that happens in an airway or a GI tract where you have all these nice stem cells that can replenish your tissues, that's not such a big deal. But when it happens in the spinal cord, we really don't think there's a lot of capacity for new neurons to form and reinnervate these muscle groups that have now lost their neurons due to death of the neurons. So, that could be a very big problem. So to really summarize this first part of the talk, the brief look into this autopsy study we did, nRVs D68 is most definitely a cause of acute flaccid myelitis, and we can see how this happens now. As I said, I think this was pretty well accepted before this study, but this was that sort of nail in the coffin. This virus definitely infects human motor neurons. It's very neurotropic, and it doesn't seem to have tropism for other cells. Maybe that macrophage, was gobbling up some infected cell debris or something, or maybe it just rarely infects macrophages. But certainly at that time point in that patient, we see a strong preference for neurotropism. And then I think this suggests that there could be multiple mechanisms at play for causing AFM. And some of those might be directly due to the virus infection of the neuron itself. And some of those might also be due to inflammatory damage due to your immune response, which, is sort of okay in some tissues, but not in a spinal cord. Where we saw these inflammatory cells. And so I'm going to pause on the pathogenesis study. I'm going to come back to that at the end of this talk. So we're going to revisit some of this at the end of the talk. And now I'm going to pivot to talking a little bit more about monoclonal antibodies. And so just as a reminder, when I was still in Vanderbilt working in Jim Crow's lab, we had isolated about 60 different human monoclonal antibodies from patients who had nRVs D68 infection, and showed that some of those antibodies can neutralize the virus quite well. I'm going to try to put that in context a little bit, what's been really nicely set up by the previous speakers, and that this is, again, that graph of incidence of AFM in the US, where we have our outbreaks in 2014, 16, and 18. all the patients who actually donated their blood where we got the B cells that we used to make the monoclonal antibodies, they were all known to be infected in 2014. They all had just respiratory disease. None of them had AFM, and they all were infected with B1 clade isolates of the virus. And then we got their, we were able to obtain their blood, actually thanks to Jim Gurn and Yuri Bochkov at the University of Wisconsin for this, around this area, so between these two outbreaks. And that's relevant because, as Emma already nicely set up for us, the types of virus, or the clades of virus that we saw circulating in 2014 that we know our patients had, that changed. And so sort of the antibodies came from here. And so any time we test for viral isolates from here, those antibodies most certainly were not generated against an infection that patient had with those isolates. And that's important because if we're thinking about developing any of these antibodies as therapeutics, we of course want those antibodies to work against any virus isolate that may happen in the future, not just the viruses that those patients had. To investigate this cross-reactivity, we ran all of our antibodies through a pretty simple in vitro screen in the laboratory bench, where we just mix different amounts of antibodies with a fixed amount of virus. We put that mixture on a cell culture monolayer, let that cook for a few days, and then stain those monolayers, and we see at higher concentrations of antibody, those cells are protected, and then as the antibody is diluted, that protection is lost. This allows us to assign a numerical value known as the half maximal effective concentration, or EC50, which is a way we just quantitate how well did this antibody neutralize this virus in the lab bench. Each of these dots is an EC50 value for an individual monoclonal antibody. About 20 of our 60 antibodies, so 1/3 of them, had some detectable neutralization against that isolate, or that clade of virus, an isolate from that clade that we know the patients had. And some of these were quite potent. So dots that are higher up on this graph are more strongly neutralizing. And these are really, really strong. Just for those who don't know antibody numbers like this very well, these are very strongly neutralizing antibodies. And when we tested against a different clade, so that A2, or also known as D clade, now we see a lot of those antibodies don't actually neutralize very well anymore. A couple do. And then when we went to that Fermon isolate, so that historical one from the 60s, only 2 of these antibodies really still had that ultra potency preserved. So this one, 228, which you're going to hear a lot more about. And then this one, sorry, I didn't label it, is known as 105. So you're going to hear about that antibody on the next slide. And now this is some newer data that hasn't been published, that was done by Xiaoyan Zhan in the Crow lab at Vanderbilt, and also contributed by Chuck Haynes from ID Biologics. What we see with these antibodies is the 105 is the top row of numbers, and the 228 is the bottom row of numbers. These are those ones that were cross-reactive when we tested them with our initial studies. We were able to really expand upon that. And these numbers, again, the lower number is a stronger neutralization. So we know our patients had B1 clade infections. These are super duper duper strong neutralizing titers for both of these antibodies. And even these numbers, although they're larger, so less strongly neutralizing, they're still pretty good numbers against this this more distantly related A clade isolates. But again, going back to that whole thing I set up, when we look at isolates from 2018, so viruses that were circulating after these antibodies had been collected and isolated, we still see a strong preservation of this really strong neutralizing activity. So that's very encouraging. Again, if we're thinking about this as an eventual therapeutic agent, either one of these, we want to make sure that those antibodies maintain that neutralization against any new clades or isolates that may emerge. So that was really exciting to see these data. And then another reason why we're pursuing them as potential therapeutic options is that they work very well in vivo models of infection. So I'll talk about this model that's from Utah State University in the lab of Bart Tarbet, and work done with Brett Hurst as well, where they have a strain of mice that they will infect. This is the AG129 mouse strain, so they're deficient in those type 1 and type 2 interferon receptors. And Dr. Freeman told us how interferon is important to these infections. When those mice are given virus intraperitoneal, and then we gave them antibodies a couple different time points after infection, we saw either paralysis in some cases or protection in other cases. And that's what I'm going to show you on the next slide. And just to, since this is older data and I've already presented it before, the take-home point from this slide is, unsurprisingly, the earlier you give a dose of antibody, the more protective it is. The longer you wait, the less protective it is. Not really terribly surprising, but what we did really see when we looked at both survival on the left and these models of these neurologic scores, where a higher score meant more paralysis in the mouse, the conclusion is the same, which is that there is a time point after, some window of opportunity after infection where the antibody can be given and can be protective. And even in some of these mice, when you really drill down into the data and stuff that's in the supplemental data of this manuscript, if you really want to find it, and I'm happy to point it out, there's cases where mice had been getting sick or had some weakness and then seemed to have maybe even gotten better. So that was very interesting. And what's, I think, super cool is that this is now not work I was involved in at all, but this is work that came and was just published a couple weeks ago out of Ken Tyler's lab at the University of Colorado, using a different mouse model. So these are actually the wrong pictures. These should be white mice now. They use an outbred Swiss Webster mouse model, and they give the virus intramuscular. So minor differences in the model, but the conclusion ends up being the same. In this case, they didn't give antibody at a particular day post-infection. They waited until the mouse was a certain amount of weak before they gave the antibody. So in this case, their sort of a weakness scale, let's call it, goes to 10 instead of 4, which I had shown you earlier. They waited until their mice had a score of 2. Then they gave a strongly neutralizing monoclonal antibody. So not our antibody, but different antibody. And what they saw was that mice with no antibody, progressed to very high scores of paralysis, and mice given antibody at whatever time point they had the onset of some weakness with this score of 2, those mice did better. And so now I'm going to try to really put all of this together, building a lot on the previous talks. So thank you again to all those speakers for setting up, some of these data. And what I've seen here, which is that, this is a hypothesis and a model, but this is how I think of how does this virus cause paralysis in humans and where are there opportunities for intervention? So, as people have discussed, there's a lot of enterovirus D68 that circulates in the world, and there's only, rare outbreaks of AFM in numbers that are much smaller than the actual numbers of infections that are happening. So the human body is pretty good at actually limiting this virus to the respiratory tract. And keeping this to a pretty innocuous infection. So for someone to get AFM, first off, there must be circulation. And Dr. Hodcroft told us a lot about, how that may change and how the virus may be always circulating, and just sort of as birth cohorts increase or other things happen, there's more virus circulation all of a sudden. The virus needs to escape the site of initial infection, which is going to typically be the respiratory tract. Dr. Freeman told us about how, interferon evasion is going to be important for that. And, there's probably other factors that allow extra permissivity in a particular host. So perhaps, we don't really fully understand receptor engagement with this virus. Perhaps there's certain polymorphism in pathways that, put extra sialic acids on the surface of cells and allow for more virus infection. And then I certainly think there has to be a scenario where there's likely no pre-existing antibodies that neutralize this virus. That's based on a lot of antibody data that we've created, but also just understanding how the poliovirus vaccines work. Those vaccines don't prevent the virus infection. I mean, they do to a degree, but they're certainly imperfect at that. But they're really good at preventing paralysis and keeping those antibodies out of your motor neurons and keeping those motor neurons healthy. So if you have circulating antibodies, I think it's very unlikely and probably impossible, if those are neutralizing antibodies, for you to get paralyzed, or at least close to impossible. So I think this is very much a vaccine preventable illness, or an illness we can prophylax against with antibodies. But once AFM onsets, then there may be sort of 2 things that drive the degree of AFM. So if Dr. Jackson's hypothesis is right, and this can disrupt neurotransmitter release by cleaving the SNAP proteins, that may be why this infection progresses so rapidly from the time of onset of weakness to the nadir, the worst point of weakness. And then also, the amount of cell death may determine just how bad the AFM gets overall and how good your chances of recovery. So I think if we can give monoclonal antibodies, we can, prevent the extent of infected cells, and then hopefully maybe give some neuromodulators that calm down that infection. So, all right, as I'm running out of time here, there are also, I just want to give this quick progress report on how we're doing on trying to get these antibodies into humans. There's really 2 groups that are leading this charge, and kind of 2 separate charges. These companies, ZabBio and KBio, have been working together to produce the 228 antibody in plants. We have a lot of, or I should say, they have a lot of material sort of ready to go at a way that it can be put into humans. We've done a lot of initial toxicity and safety studies, and really, actually, just last week we pitched to NIAID to try to get a phase 1 trial going for this antibody. And then also the company ID Biologics I mentioned before is working on still these sort of 2 candidate antibodies to produce them in what is a more traditional way of making monoclonal antibodies. And right now, just kind of determining which one is the best one to move forward with from scalability perspective. And NIAID has been absolutely instrumental in moving these along, because trying to study therapeutics for rare diseases, drug companies aren't looking to spend a lot of money on those things. And so the role of NIAID cannot be undersold. And they've contributed in a lot of different ways, a lot of different funding mechanisms, including their preclinical services division, SBIR to ID Biologics, and some funding to us in the Crow Lab, and different divisions of NIAID have contributed as well. So, all right, I need to stop talking and need to move on to some questions. So let's do that.
Thank you so much, Matt, for that great presentation. And I will field some of the questions from the audience for you. There are already several questions. The first is, are autopsy similar findings seen in the wild type or in the CVD polio cases?
Yeah. So it's interesting because the technology has changed so much since when poliovirus was the major circulating cause of AFM. Because to be clear, AFM is an umbrella term, and poliomyelitis is just a version of that caused by poliovirus. And then it has some of its own clinical findings that are a little different. But when we think about that patient in New York who just had the CVDPV, version of poliomyelitis, they were identified through the AFM identification system that we have, that the CDC has set up, and because it really is just, you can't tell when someone's in front of you until you find out which virus they have, which version they have. So yes, so I think my understanding is grossly motor neuron loss in the anterior horn of the spinal cord is what is the end result of both poliovirus infection and enterovirus D68 infection that results in AFM. But to be clear, not all infections result in that. In fact, it's the minority of infections that result in that. But yeah, that is seen.
Thank you, Matt. So what are your thoughts on a study showing potential for retrograde axonal transport and potential for a spread directly from the upper respiratory tract to the central nervous system, perhaps via olfactory route? What are the implications for monoclonal antibody therapies if the virus is never in blood? Do we need mucosal vaccines or IgA antibodies?
Yeah. So interestingly enough, that can also happen, with poliovirus. The vaccine can spread directly from skeletal muscle, cross that neuromuscular junction, and go right into those, those motor neurons in the spinal cord. And yet, circulating antibodies prevent poliomyelitis quite well. The mucosal vaccine for poliovirus, which is the oral poliovirus vaccine, is better at limiting transmission. And I would suspect that a mucosal vaccine for enterovirus D68 would be better at limiting transmission as well. I think there's certainly a role for these potential things. But I think it might be as simple as people having circulating IgG that neutralizes the virus, protects that spinal cord. But we don't know, I think, is the answer. I mean, it's hard to really say in a human being, is the virus capable of going directly from the lungs to the spinal cord? We can do a lot of stuff in mice, and believe me, we're pursuing studies in mice to try to understand these questions, but I don't think we have that particular question answered just yet.